Welcome to The Edge, a program where we spotlight Purdue University scholars and innovators, highlighting their careers, scientific achievements, and their vision for the future of their scientific field. My name is Tommy Source. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And on this segment, we will be focusing on microbiome sciences. I have the pleasure of welcoming Dr. Caitlin Proctor, who is Assistant Professor in the Department of Agriculture and Biological Engineering and Environmental and Ecological Engineering. Thank you so much for joining us today, Caitlin, and welcome to the program.
I'm so happy to be here. Thanks for having me.
Well, I am so excited to learn more about your background and the work that you're doing in your field. You have a very unique research program, and it's something that I really want people to hear about. And so I've been looking forward to interviewing you and picking your brain, if we can, a little bit as to where are we going in your field. So Caitlin, maybe you can start us off by giving us a brief background of your education and your training and all of that stuff. So get us started.
All right. So I started off in civil engineering at Virginia Tech. And when I graduated, I thought I wanted to get a job in industry, but it was a recession. So I decided to get a master's in environmental engineering. Was really excited to do that. I worked with Mark Edwards and Amy Pruden at Virginia Tech. And from there, during my master's, found out about this amazing PhD program in Switzerland. Had no intention of getting a PhD, but thought, why not go to Switzerland, where I got to do a PhD in life sciences, focusing on microbiology. From there, I found out about the Lillian Gilbreth postdoctoral fellowship here at Purdue, and I got an opportunity to stay on as a faculty after that.
That's awesome. I mean, you say that so quickly as if it just happened so fast and so effortlessly. I think it's quite a feat. In looking a little bit at your CV, I did a little bit of research, and you've been really focused not only on microbiology, but a very specific area, which is, water quality and all everything that has to deal with our infrastructure and how we connect to the water that we consume. And it's just fascinating, number one, to see how dedicated you have been to that field for such a long time. Can you tell us a little bit, like, what was the first inspiration for getting into that, and like how did you go about that?
Yeah. So, first getting into it, like a lot of things in my career, was a little bit by accident. My research advisor for my master's gave me two options for projects. One was in wastewater and one was in drinking water. And the wastewater lab smelled bad. So I decided to take the drinking water project. And with that, I learned a lot about, so Legionella is one of the main pathogens that lives in drinking water systems, Legionella pneumophila to be more specific. And I was just so fascinated that organisms can live in this environment that we think is so harsh, and all of the things that we can do as engineers, as water users, to either allow this pathogen to grow or keep it from growing. And I've just been, fascinated by not just this pathogen, but all of the other organisms that live in these systems that are designed to not have anything living in them. But these organisms persist, and it's been really interesting to dig into why and how and what we can do to prevent that, and also to prevent other water quality issues that happen in the same locations.
And that's fascinating, because we've heard in the news more recently about water quality, but it's been more on the contamination with heavy metals like lead, right? We've heard of incidences, but you're more focused now on the microbial side of things and how things are changing dynamically. I know you've recently published on how things change when you have stagnation in buildings like the ones we had during COVID. Tell us a little bit about this, Caitlin, if you can.
Yeah, so water is, distributed with a chlorine residual that's designed to prevent organisms from growing. So we want to prevent things like E. coli that we know are bad in the water from growing. And by doing this chlorination that we've done for so long in the US, we've really prevented a lot of disease. But in recent years, we've discovered that organisms also grow in these pipes. They're not the same type of organisms as these E. coli that like a lot of nutrients. They just persist in these biofilms along the walls of the pipes. And so when water sits for a long time inside buildings, there's not chlorine that's constantly moving through. So these organisms can grow and proliferate and get into the water. And from there, if you inhale those organisms, they can cause a lung infection that can eventually be fatal, especially with immunocompromised persons. So these biofilms exist within our plumbing system no matter what, but we were really worried when the COVID-19 shutdowns happened, what would happen to these buildings that went completely unused. Now, surprisingly, another thing that happens when chlorine isn't being introduced repeatedly to these systems to stress the microorganisms out, we also have a flip side where nutrients are also not being introduced. So a lot of the growth issues that we were really worried about were not a problem in the buildings that had very little to no use, but more a problem in the buildings that had little to moderate use. So it's been a really interesting journey to look at, the entire microbiome, the entire ecosystem living inside these pipes, and how they react to the changes in how we use them.
That is, again, so such an eye opener, because you end up showing the importance of how we should be dealing with water as as our source in our office buildings, in our homes, in our and Caitlin, tell us a little bit about what's the regulatory landscape look like here? Is it like very strict? Who, first of all, tell us a little bit, what are we seeing, and how is that changing in this type of environment, especially with the work that you're doing?
Yeah, so it's really tough to regulate water quality in such a big system. So to think about the miles and miles of pipe that are between you as a water user and your drinking water treatment plant, a lot of changes can happen over that space. So regulating things that happen within the distribution system can be really difficult. So whether that's lead and copper coming into the pipes out of the pipes and into the water, or these microorganisms that grow, it's really hard to know where these reactions are going to happen and when, and controlling them is difficult. So the utility is responsible for water up until the property line, but after that, the building owner is responsible. So there's not much regulation for the building owner. That would be really difficult. You'd think, anyone who owns a building has to become a water quality expert, and that would be pretty difficult to maintain. But buildings with people who are at higher risk, like hospitals and nursing facilities, have a little bit more regulation and a little bit more control. So they will have things like testing programs to look at the growth that's happening in the water. But buildings like office buildings or your gyms or your big apartment buildings are not going to have the same kind of regulations for the water that's going to the tap.
And so what's happening in the field? Is the technology getting better for these places to start monitoring, or are you getting busier and busier because people are like, oh my gosh, I need Dr. Proctor to come and help me understand where we should be collecting, what we should be collecting, how frequently, and all of this stuff? I mean, where is that horizon, Caitlin?
It's difficult because it is evolving quickly, and there's questions about what do we do if we do find these organisms, because if people haven't gotten sick, then maybe it's not the biggest deal in the world. So I think it really depends on the building owner and how much work they want to put into protecting the people using their water. There are things called building water management plans, where even if people just take a look at their water system, understand where their pipes are going, understand things like how hot their water heater is, and that's going to have a huge impact on the type of organisms that are going to grow. So building water management plans are a great way to go before things are regulated and to protect people. A lot of consulting companies are popping up, more and more consulting companies, to create these for people and help them understand their water. But there's a balance, there's this risk, but how many people are really getting sick? How much money and effort can we put into this? It's a balancing act for sure.
Yeah, I can imagine. And the conversation is probably dynamically changing every day and evolving too. If you were to think 5 years, 10 years down the line, where do you want to take the field? What is it that you want to do with it?
Yeah, so I think there's this fear right now because the organisms that we hear about are pathogens that can potentially kill people, and no one wants that. No one wants that out of the water coming out of their tap. But I think we can move forward a bit past that to just understand that there are organisms growing in these systems no matter what. And I think there's ways to work with the organisms that are growing to, promote beneficial biofilms that maybe even prevent pathogens. But the idea of, putting microbes into a system right now, no one's on board with that. The idea of limiting the amount of chlorine we use so that we could go towards a more beneficial biofilm route, no one's really on board with that. But I'd love for people to understand that, yeah, there's microbes in your water. It's not a sterile water system, but it's okay. And it's not everything is out to kill you. And there are ways to, work with it.
And are you developing new ways of looking at the water quality in your lab? What type of work is done in your lab?
Yeah, so I think, we're trying to understand what microbes are growing, why they're growing, what they're eating, what they're growing on. And I think, the more we understand about these ecosystems and how they function down to a really, basal step, we're going to be able to manipulate those biofilms and manipulate those microbes. So, looking at pipe material, how can the type of pipes we put into our system change the microbes that are growing? How can we think about, flushing? Could we redesign our plumbing system so that water isn't sitting still for so long, so that it's automatically circulating, so that we, put the dirtier water towards dirtier uses like irrigation that might have less of a chance of infecting people. So I think there's a lot of things that we can do. I'm looking at how can we redesign plumbing? How can we redesign the biofilms that are there? How might synthetic biology be useful in this scenario? I think there's a lot of different ways to go.
So neat. It's so awesome. And it seems like a field that is just exploding, right? Like there is so many questions still to explore. What is, and here, Caitlin, I call this program The Edge because I want people to know that what you're doing is really the edge of knowledge, the edge of this technology advancement in this field of microbiome for water management, for water safety. What's one myth that you want to debunk in this field? What is it that you want people to know? And I think you've told us a few tidbits here and there, but what's a big one?
So I think the biggest one that I'm surprised, I'll go in, I'll even talk to doctors who know about diseases, who know about microbes, And they will think that the water, drinking water that comes out of the tap is sterile, that there is nothing growing in there, that it's completely clean, that the chlorine takes care of every possible issue. And that's just not the case. You know, there's microbes everywhere. There's microbes on your cell phone, there's microbes on your hands, there's microbes in your water. And I think the more we accept that, the more we accept the microbes all around us, the less fear we might have about, you know, how to deal with them.
Yeah, it seems like microbe warfare out there, and we're just vessels, right? Carrying them around, and we're probably like the best vessels for them to reproduce on, to pass on genes, right?
Oh yeah, and we do things that aren't great. Like we pump a lot of antibiotics into the environment so that the microbes are fueled and they can develop antibiotic resistance. And of course, if these genes get into the pathogens that infect us, then going to the doctor is that much harder. Our antibiotics aren't working. Even, you know, people have been looking at the relationship between the use of chlorine and antibiotic resistance. You know, by stressing these microbes out, are we selecting for the toughest microbes for us to be exposed to? It's probably not a good thing. You know, we don't want to escalate this warfare. We want to work with them as much as we can so that we can fight them when we need to, but not all battles need to be fought.
Right, right. And is it something that it requires time and education, you think? Or is this something that really we're going to need to start showing more and more evidence that is going to convince people? Where are we at with that?
It's so hard to say. You know, we've been hearing about the dangers of, for example, antibiotic resistant microbes for so long, and still people are using Purell all the time, even if they don't need to. Everything has an antibiotic built in. And, you know, there's evidence that this is increasing our disease burden, making it harder to treat things and killing more people ultimately. So I'm not sure what the next step is, but I do think education plays a part of it. You know, every time I do a presentation, I scare people about taking showers, but I hope that by the end of it, they, you know, understand that, oh, it's going to be okay. Like, not all showers are dangerous. I've been doing this every day and I haven't gotten sick yet. It might be okay. So I think, you know,
Okay.
education takes a lot of different forms.
Well, so let's use this platform maybe just to dispel that. I think I know the answer, but please explain to us, why is the shower a potential source for us to be infected with a microbe from the water?
Yeah, so a lot of the microbes that I work with are respiratory pathogens. So the way that they're going to act on you is that you might inhale them, and then they would essentially form a biofilm on your lungs. And of course, that's not where you want a biofilm. So, you know, every time you're in the shower, there are microbes and aerosols all around you, and you are inhaling a little bit of that and getting it into your body. So definitely you're exposed to microbes, even when you're cleaning microbes off, you know, so it's just the way it is.
So you're taking a nice hot shower, and the steam that's coming out of the shower, you're inhaling that?
Yeah, so water droplets, aerosols, you know, small droplets you're inhaling that, and some of that goes into your lungs. There's also exposure as you drink water, you're going to aspirate a little bit of that, and it's going to go into your lungs. You're also exposed to microbes that might infect your wounds. So if you have an open cut, that can be infected. So, you know, for a lot of people, you're able to fight that off. You have an immune system for a reason that's able to attack these microbes that are coming in. But it's more of a problem for people who are immunocompromised, people who are sick already, people who are taking immunosuppressing drugs, for example. And then they get exposed to these microbes in the same way, and their body can't fight them off.
So that, and again, like, my gosh, I hope that's an eye opener for people as well. You mentioned gyms before, and I want to go back to this whole first piece of work that you did with Legionella, and like how does that fit in with gyms and jacuzzis? We always hear about, maybe spas and spa treatments could be a danger for picking up Legionella. How do they even know? Are there restrictions? How does that work, Caitlin?
Yeah, so my parents have a jacuzzi in their backyard, and I am on them all the time that they have to chlorinate that, they have to treat the water, they have to keep it at temperature, because if their temperatures are too low, then microbes are going to grow in that hot water. If there's not enough chlorine, microbes are going to grow. And then again, just like in the shower where there's aerosolization of that water, where there's, vapors, not exactly vapors, but small water droplets, you're going to inhale some of that. So there's been some major outbreaks of Legionnaire's disease, for example, at Jacuzzi. I think that there was one at a jacuzzi tub show where like they just had hundreds of jacuzzi tubs. There's been outbreaks at flower shows where they have a lot of those misters that are spraying water droplets down. But the same thing's going to happen in a spa, in a steam room, if that water is not clean enough. And then for, gyms, you don't know the last time that shower has been used. If it's in a certain type of use pattern where it's used somewhat, but not too often, that might be a hot spot where Legionella or other organisms are growing.
Interesting. So interesting. And I think we should be careful because I think jacuzzi is a brand name. We should be referring to them as hot tubs, right?
Sorry. Sorry,
Me
yes.
too. No, me too. I think I was the first to say, oh, in the jacuzzi, but really it's not just jacuzzis, it's any hot tub, any source of warm or hot, what we're calling hot tub, but it's really not hot. It's warm-ish water that
And
we
it grows in cold water too. So, some of this has been linked to misters at amusement parks or gardening, for example, or greenhouse misters. Anything that's going to create a water aerosol is a potential route of exposure.
Right. And what about these natural springs? Is the risk as high in those places as well, where they're natural springs?
I'm not sure I've seen anything about springs per se, but certainly in the environment, there are ways to be exposed to microbes. So, swimming in a natural lake, you're going to be exposed to Legionella, but also maybe things like Naegleria fowleri, which is the brain-eating amoeba. So there's all sorts of microbes everywhere you go. One thing I really like to harp down on people is to never use nasal flushes, and I don't want to say the brand name of that, but if you do use a nasal flush where you're going to, stick water up right next to your blood-brain barrier, you're going to want to make sure that that water is clean. So people can get exposed that way, but also slip and slides or jumping into the water. There's lots of ways for that water with tons of microbes in it to get into your body in ways that it's harder to fight off.
Yeah, that's such a great warning piece there, because I think people need to be more aware, and by creating that kind of awareness, hopefully people will be safer, right? And they'll be able to keep their kids safer, themselves safer, their families safer.
And it has nothing. One thing I might want to emphasize is that has nothing to do with not trusting the water that's coming out. Like, everything can be perfectly right with that water, but the temperatures inside your home or the biofilms are growing or your hose in your backyard hasn't been used for a while. So it's just a matter of making sure that you're using that water for the uses that it was intended. No drinking water is going to be treated such that it's advisable to stick it up your nose next to your brain. There's just no way to treat it that well.
Right. No, interesting. Tell us a little bit about these. In my house, I have a charcoal filter, and then I also have like a UV light filter, it makes the water taste really, really good. Like it tastes really fantastic. Is it because I'm removing, I'm taking away stuff, right? With that charcoal filter, right?
Yeah, so charcoal filters or activated carbon filters are going to remove a lot of different components in your water. One of the main things that you might be tasting the difference of is that it removes chlorine. So it's going to remove that kind of pool water taste that you might not like. And certainly people who are in Europe where they don't chlorinate the water really hate that chlorine taste. So
Yeah,
you're removing what's protecting the water against growth.
right.
Um,
So, but I worry, Caitlin, past the filter,
yeah.
after the filter, there's this water spout that comes up, and I fill my water with that. Should I be concerned to flush out that after filter tube and make sure that there's nothing growing there? Because past that, past the filter, there's no chlorine anymore. So stuff could be growing there, isn't that right?
Oh yeah, I guarantee you stuff is growing there. And it's probably growing upstream too, because there's not going to be really high levels of chlorine that are delivered to your home all the time either. So there's stuff growing everywhere. I think it's just a matter of thinking about how you use the water. Again, I would not take that water, the first flush of that water, and stick it up my nose, but
Yeah,
I might be okay drinking it if I'm a healthy adult. So
sure. Okay. Uh, One question that you wish people asked you more that people never ask you. I'm giving you carte blanche, open stage here.
yeah, so, I hope I've reassured people a little bit about, their infrastructure isn't terrible, even though I'm pointing out problems with it. But I do think, thinking about the future of infrastructure is an important question that I love to get asked about. Like, what is the future of plumbing? How is it going to, change based on all that we know? And there's a lot of things that might need to change or have to change. We designed pipes for a really large diameter because all of our appliances needed that. As those diameters decrease, what else can we be changing about our plumbing system to match up with the appliances that we're using, the way that we're using water? So I think there's a lot of potential for redesign in plumbing infrastructure, as well as drinking water infrastructure at large. So thinking more about the different ways that we use water, having different levels of treatment for that could be in our future. For example, maybe gray water can be used for certain activities, but we want to keep our clean water for other activities. So I think there's a lot we can do with the future of plumbing and redesigning that for our future needs.
And that is so awesome. And being at Purdue with all of the engineers around you, I mean, this must be a huge playground, and what an opportunity to delve not only into the engineering of the infrastructure, but materials engineering and engineering, the future of contained and self-contained systems. And like people are talking about going to space and living in
Yeah.
the moon and things and Mars, and like how are you going to deal with the water there? And that it must be a big playground for you.
Absolutely. And there's so many, partnerships I've been able to develop, thinking about, like you said, NASA, they have issues with the growth in their plumbing systems. As much as they control every little thing that happens that goes into space, there are microbes in their drinking water system. They have biofilms on their pipes that they are trying to figure out what to do with. So I think the future there might be to work with the microbes, not completely against them, because if NASA can't keep microbes out of water, I don't know who can.
That's so true, right? Okay, like they're supposed to be able to do a lot of things. Tell us a little bit about the material side. I know things have changing because I saw even my plumber, he changed a pipe that was copper and he just put PVC, And I saw that and I said, oh, how interesting. Copper must be pretty expensive. But is that the only reason, or is there some advantage to PVC that copper didn't have, other than, it's easier to, screw on or whatever design?
Yeah, so the how plastics impact plumbing has been a really interesting place that I've been able to explore. Even one PVC is going to be different than another PVC in terms of the linkage that's occurring, in terms of the carbon that can leach out. And that can have an effect on the microbes that are going to grow in that space, because that's a major carbon source, a major food source for the microbes coming into the system. So I think there's a lot to explore with respect to, which plastics are we putting into our water systems. There are some that don't leach a lot of carbon. These tend to be more expensive, some that do leach a lot of carbon. So that absolutely remains to be explored, and something that I'm looking into in collaboration with people in material science. As far as, replacement from copper to PVC, that's definitely a trend in plumbing. And it is a lot driven by money and ease. But the problem is that we don't necessarily know the long-term effects of that. So, PVC has really only been popular in plumbing for a few years compared to copper. So looking at those long-term effects is something that I'm interested in as well.
Yeah, and I would think, copper not only being expensive, but those pipes thin out after years of usage, right? So as a material, having a metal that continuously sloughing off, I mean, might be good as an antimicrobial, but as a way of, sustaining that water for the long time under pressure is difficult, right, to think of.
Yeah, so I mean, so you mentioned copper being antimicrobial, and I guarantee you, if you find an old copper pipe, that antimicrobial effect wears off eventually. It definitely becomes a home for biofilm at some point. But sure, there have been plenty of issues with, leaching of copper into water, and copper is, a gastrointestinal irritant, so that can cause problems as well. And, how long do any of these pipes last? I'm not sure that we know completely. So in homes, plumbing gets replaced every now and again when you redo your kitchen, redo your bathroom, have a leak. So there's definitely a lifespan to all of this. One way that's really interesting that they've been dealing with it in the drinking water system, like out in the distribution system, is to actually take the old pipes and line them with new plastic. So they can, in place, add another layer on the inside. But these layers are plastics that are being formed in place. So like they'll line a pipe with epoxy, for example. And one of my students right now is looking into, what does that mean? Is that new pipe always going to be perfectly made? Is that going to provide a lot of food for bacteria? Are these ways of dealing with our crumbling infrastructure going to create more problems in the end? So there's some really creative solutions out there, but the long-term effects have yet to be fully determined.
Well, and how can you also put so much pressure on the manufacturing side to change the way that we're going to be manufacturing these things too, right? And is that really sustainable, or is that really a way of leading the change, right? It's out there. And what I love about what we do at the university is always push that boundary, push the edge, right? And I really like that concept that, you guys are trying different materials, different ways of sampling. Uh, Tell us about, like, for the future of infrastructure. I mean, like, would they call you and say, "Hey, we're thinking about forming a new building over there, and we would like it to be as safe as possible for water"? Would that be something that they would call you to be a consultant on a project?
Yeah, I think so. And I wish it were more a part of the design process, because there's a lot of little things that can be done. For example, if you think about your home, how many bathrooms do you have? How spread out are they in the house? That is all creating more pipe, more places for issues to happen, more points for failure. So thinking about that in the design process to, if you have bathrooms on every floor, stacking them exactly on top of each other, so there's not as much branching pipe. And then you can add, recirculation systems. So for hot water in large buildings, rather than having one big tank and a line that goes out that cools down every time you're not using it, you can recirculate that water to maintain high temperatures to the point of use or very close to the point of use. So there's absolutely ways to make infrastructure safer. And it does need to be considered more and more in the design phase. And I think there's more creative solutions that we can go towards beyond what we know now. So I'd love for that to be happening in the design phase. It's often an afterthought, architects don't necessarily think about that. They think about the flow of the house and how to make it convenient for everyone. And
Sure.
but one thing I really rail against is, these big houses with, bars in the basement, and the bar gets used once a year for the holiday party. Like, do you really need a sink there? Or is that just extra pipe that's dead space for things to happen, space for reactions and growth that doesn't need to be there?
Right. Oh, how interesting. So neat. I'm sure there are a lot of people that are thinking about that and saying, oh, you're right. or that bathroom that never gets used, right? On the corner or the basement somewhere.
Yeah, and I mean, never using it might be better than trying to use it like once a week, because then you get into that situation where you're bringing new nutrients in, but
Yep.
just taking it out is going to be a better solution altogether.
Yeah. Wow. Again, such an eye opener. Caitlin, if you were to look at the research that you do and you look at the field, what are certain gaps that still remain that you think we can apply solutions to, or that we can innovate towards?
Yeah, so I think my field is largely focused on this one pathogen, Legionella pneumophila. That's where a lot of the research money goes. A lot of the infrastructure is trying to, get rid of this one pathogen. But there's more and more evidence that by getting rid of this one pathogen, we're making space, making niches open up for other pathogens. So I think, the field is moving towards this more holistic approach where we're going to look at all of the microbes that are growing and trying to understand not just who is there, but why are they there? What are they eating? How are they interacting with other microbes? And it's really an exciting time to be able to explore all of these things. So there is still a lot more to explore because, everything that I learned about this one ecosystem in one building might not apply in another building, in another ecosystem. So understanding that true diversity is something that's been really exciting and kind of the leading edge.
Well, and because of I get to see so many and talk to so many different scientists like you, I get to know a little bit about what is happening in the field. What makes it most exciting for you? Because I know what's most exciting for me, but what makes it more exciting for you?
I think just, illuminating everything that's happening inside these pipes and being able to talk to people has been really exciting as I've gone. And there's just so much more with, all these new methods that are being developed and them getting cheaper and cheaper. Like we can analyze more samples, we can understand more of these dynamics, know at a tighter time scale, not just, 1 month to month 1 to month 2, but week 1 to week 2 or day 1 to day 2, what's changing in these systems. So the methods I've been developing so quickly to help us broaden our, knowledge of all of these things. Drinking water, we weren't able to study it in this way for a long time because there simply wasn't enough biomass and the methods weren't designed for these low biomass environments. So just all of these methods and how fast they're developing and how much we can use bioinformatics and, working with data scientists because we're getting so much data that we need data scientists. It's exciting.
That's awesome. And I do agree. I think that's exactly where I was going with this is the amount of data that we can pull out from the samples that we were collecting even before and know what was there, right? Tell me a little bit about genomics sequencing and like the bioinformatics that you spoke about. Tell me a little bit about like how is that opening your field? What's happening there?
Yeah, so, I think when I was getting started, this thing called amplicon sequencing was starting to take over. And with this, we can take a look at like tiny little snapshots of all the organisms that are present in an environment. Before this, we had to grow all of our organisms and have enough of that biomass to be able to send it out to see the genetic sequences that were present, see the different, genes and functions and niches that these organisms could occupy. But, going from, sorry, we couldn't culture, we couldn't grow a lot of the organisms in drinking water. So many of the organisms in drinking water are so slow growing or so low nutrient that getting them to grow on a plate was just impossible. So being able to, first look at, all right, we can at least say who is there was really cool. And then, you get thousands of sequences and, gigabytes of data. But now we're, moving to longer sequences to not only be able to say like, we think this is this organism, but we know this is this organism. Being able to look at the metagenome. So that's being able to look not just at like one specific sequence for taxonomy identification, but also being able to say, these are the genes that are being expressed. These are the metabolisms that are involved. And there's just so many more methods, proteomics and everything that we can be able to just look at more and more detail to answer different kinds of questions about what's happening.
And I'm so amazed at how we are starting to synthesize all of this information, right? And you said it best. You are illuminating what's in these pipes in a way that, and I'm sure companies like Illumina are happy to hear that too, right? That their sequencers are helping us really open up the light and see what's happening inside these pipes in a exquisite way. So what you're saying is that not only can we find out who's there, but also we can start to analyze what they're doing in there. Can we also know how many of them there are compared to others? Can we do numbers?
Yeah, so it's growing more and more. So, see like looking at who's there, we often don't get total abundance, we'll get relative abundance. So we can say, 1% of these sequences look like they were from this organism and so on. We can follow that up with, designing specific assays, qPCR assays to quantify exactly how many of that organism are in the system. So we can fill in that analysis. But more of the analysis is also going to not just this one gene, but looking at all the different genes and what they're doing together. So we're getting more and more data, more numeric data, being able to combine some of this data. It's all going in databases that are making some really cool meta-analyses possible. So it's only growing.
So the tools are getting better, is what you're saying, right?
And more accessible. So, cheaper and being able to run more things. When I first started, to be able to have 10 sequencing samples in a paper was amazing. Now you need hundreds to be able to be publishable. So the field is changing because it's getting cheaper and it's getting like you have to have more resolution to be able to be novel and exciting.
But I'm sure there's a huge opportunity there as the technologies are getting better, but also as the questions that we can start asking become more apparent, right? What are some of those things? I mean, I can only imagine the questions that people are starting to dig into. But so in the world of Legionella and water safety, as you said before, is it looking for other microbes that might be competing against Legionella, but they might not be harmful for us to consume? Is that the way you're thinking about this?
Yeah, and there's a few researchers that are looking into this same question. Like, if we were to design a biofilm that, Legionella cannot invade, a biofilm that we consider to be safe, considered to not have human health consequences, who would be there? What would they be doing? So we can look, not only at occurrence data to say, when Legionella is present, this one isn't, or when Legionella is not present, this one is here, so maybe they're a competitor. But it's going to take a lot to go the next step to prove all these, correlations. Proving causation is going to be another step that we need to get towards. And then we're going to have to, isolate more microbes, experiment with them together, do growth assays. So we're moving from this kind of correlative state to causation. And that's an important trend in any science field, I think.
Yeah, absolutely. And I'm so excited to see what's coming next in your field, because again, there's so many opportunities to continue to explore and to fill certain gaps that you've demonstrated, you've told us about today. It's so wonderful. I really I appreciate you taking time to reveal some of this to us, because I'm sure not too many people think about this. If you were to go back and you were to start all over again, would you do the same things that you did? Would you follow the same trajectory? And maybe more fair question, Caitlin, is if somebody were to think about a career similar to the way that you have, what should they be thinking about? And I know this is selfish. I do this for students, for people who are thinking about heading in this direction.
Yeah, so I think it's hard for me to give that advice. And I have, student advisees come in and, ask like, how did you get there? And every step of the way was a little bit of an accident, just happened to be at the right place at the right time, hear about the right thing. So I think, it's okay to not have a perfect plan is the advice that I would give. And tell myself, my younger self, to embrace that lack of a plan, because opportunities are going to present themselves. Not that you can do nothing and they'll just appear, but if you look for them in the right places, look for things in the places you want to go, the people you want to work with, you're going to find opportunities that are worth pursuing.
Awesome. And what would you say in terms of preparation? How can they best prepare for, the type of work that you do? I know obviously you need to do a lot of grant writing, so writing and communicating are important, but what else? And sorry.
No, sorry, just grabbing some water.
Yeah.
Yeah, I think, giving it everything you have along every step of the way, even if, you're, finding every opportunity and just giving it your all is going to be helpful to get you to that next step. So if you're in one research lab, even if it's not the lab you want to be in, give it everything, get that amazing recommendation letter so that you can move on to another lab that you're interested in. Read everything, try to embrace all of the exciting things around you. The more you know, the more you're curious, and that's super important for being a researcher. Absolutely, writing and communication skills. I can't tell you the number of hours I spent perfecting my PowerPoints, and I'm glad that I did, because that, being able to communicate your research is just as important as the research itself. So, all of these soft skills are going to be important in going forward with research. It's not just about how fast you can pipette and how good you are with R. It's about all of these things around it as well.
And so if somebody wanted to, join your lab as a grad student, as a postdoc, what are certain areas of strength that they should have?
Yeah, so for, grad students, I look for a prior interest in research. So I really encourage students, if you want to go to grad school, make sure that you've done summer research experiences, worked in a lab to be able to demonstrate that you have some of those skills is going to be really important. Honestly, the people working in my lab come from all kinds of different backgrounds. I've got civil engineers, biological engineers. So where you're coming from doesn't really matter, but if you've got a passion for the topic, that I think is going to be one of the most important drivers for success, especially in my lab. So having a passion for clean drinking water, for that communication piece, as well as the science of it, that's going to help you stand out.
And what about collaborators? If you were to say, what would be the types of collaborators you would be looking for now?
Yeah, so I think, enthusiastic people, same thing for working in my lab. It doesn't really matter what you're working on, but having that enthusiasm is going to be so important for me to, I like to work with people I have fun with. So, if I'm able to have a good conversation about research, I'm sure we can find something to work on together. And that's been so lovely at Purdue. There's just so many different people to work with, all the different departments. It's been really amazing.
That's awesome. Can you give us a few of the areas that complement your areas of expertise, and like that, if there are people that are watching, they could be like, oh, maybe I could be a collaborator to Dr. Proctor.
Yeah, so I'm working with people in mechanical engineering who are doing water treatment things, like for example, reverse osmosis treatment. Material scientists, because material is going to play such an important role in biofilm that's forming there. I've got some collaborative projects going in the hydroponic space, because that's a controlled infrastructure with water, similar to the drinking water infrastructure. So I've really loved kind of going into that slightly different environment, but there's a lot of parallels. But really, if you've got any microbial issues or microbial questions, no matter what the system, there's going to be a way to engineer those microbes, and I'm going to be interested in collaborating them.
I'm so glad you mentioned that, because I do know that Indiana has a growing aquaculture industry, and we're farming shrimp here, and we're farming fish, and I'm sure that's a huge area where people are probably going to be calling you more often. Is that been the case?
Yeah, no, and there's also people who specialize in these different systems. I'm just looking to, help and complement those skills. But yeah, I've been, working with some growers in Indiana where they're worried about microbes, because microbes can come in, whether they're food pathogens or something that will, kill the crop or be harmful to humans. It's something that they want to keep out of their system, and knowing how to do that and understanding how to do that requires this more holistic, complete understanding of which microbes are already growing there, why are they growing there. So I'm really interested in all of those types of things, just to keep people safe, keep our crops safe, keep our animals safe. All important things.
Absolutely. Absolutely. And it's a way to, it's a perspective, right? It's a philosophy that you have, that you want people to think about, and use this. Again, it's been such an eye opener, Caitlin. I thank you so much for using that time to give us some of this. I'll ask you, can you name us, 2, 3 mentors in your life, and maybe some advice that you've gotten from them that you want to pass on to the people that are watching today?
Yeah, so I think, working in the drinking water industry has been really interesting. It can be an old boys club at time, but there's been some really amazing female mentors that I've had, like Michelle Prevost in Canada, Amy Pruden, who was my advisor at Virginia Tech, and Jennifer Clancy, who's more in industry. And just seeing all of these women just excel and, go forward and have confidence with, I'm right, even if the old boys club doesn't agree with me, this is the way that it has to be. So I think seeing them lead by example and being strong and assertive, and if you've got the right science, the rest is going to follow. So I've been really in awe of all of them, really, for being pioneers.
Fantastic. And what a great way to put it also. And for you to say that here in Indiana, that there is great work for female engineers, female scientists, people that want to do good work with you, the opportunity is open. And I really like that, that you reflect back on a few of these people that have given you, strength and encouragement to move forward. Caitlin, thank you so much for doing this. Do you have any parting words for the people that are watching? Anything that we have not covered today that you want to cover?
No, I think we've covered so much. Just, I think microbes are everywhere might be the last thing, and it's not something to be afraid of, even if I did have some scary thoughts in there.
Awesome. Well, thank you so much, Dr. Caitlin Proctor, Assistant Professor in the Department of Agriculture and Biological Engineering and the Department of Environmental and Ecological Engineering. Thank you for sharing with us, and I hope you have a great afternoon.
Thanks, you too. Bye.
Bye-bye.